Hello, this is Patel Farin Yasin, and today I am going to explain insulin gene with PTE vector based on PCR process. So basically I took insulin gene. Now I am focusing on the main functions of the insulin gene. So this gene encodes the insulin and regulate the glucose hematostasis with disruptions linked to metabolic disorder including diabetes mellitus. Now we can see this kind of figure. This is a simple figure with synthesis of insulin in three different kind of tissues from pancreas such as liver, skeletal muscles and adipose tissue. Now, this is also another mechanism actions of the insulin, which involves a series of cellular and molecular process that leads to productions and secretions of insulin in liver. So now this is a 9 basic steps of transfer and cloning of the insulin gene. So we start from the beginning. So the very first step is gene isolations. In this step, we can extract the insulin gene from human or any kind of animals. Now we going to the next step. This is a vector insertions. So basically we can insert insulin gene into the cloning vectors such as plasmid or viral vectors. Now the third step is a transformation, so we can introduce recombinant vectors. Now the another step is selections and replications, so we are identify transfer gene in these steps. Now the another step is gene expressions and inductions. Next is the protein expressions, purifications. So in these purifications we can extract purified insulin gene. Now the next step is a verifications. So in these verifications, we can identify and confirm the functions of the clone insulin. And the last step is a scale up. So in this step, we can see a sufficient insulin yield. So this is a basic information which involve a transfer and cloning of the insulin gene. So now I am explaining what is the actual process of cloning of insulin gene. So this is a main strategy of synthesis of insulin. So first of all, we can extract mRNA for insulin in beta cell which is present in a pancreas, and in this next step based on the PCR process, RNA convert into the DNA. DNA basically we know in this PCR process always RNA convert into the DNA. Now in the next step they produce double standard DNA in a plasmid. Now in this next step they transfer recombinant DNA into the bacteria, and in this last step insulin synthesized from the recombinant DNA and it will be purified. So, this is a main strategy of synthesis of insulin uh, based on the PCR process. Now, this is also This is also a figure which clarify two different kind of step. This is a simple This is a same figure, but I put it for a clarifications, divided into T-step transfer of the insulin and cloning the insulin gene. So this is for a clarifications from synthesis of insulin gene. Now we are focusing on quality control and validation for insulin gene for expression vector. QC validation QC validation for insulin gene expressions PTE vector includes several key steps to ensure the accuracy and functionality of express insulin gene. So basically this is my slide which I explained nicely.